Welcome to IFERP, a forum where innovations and research interest could be supported and developed, prioritizing our mutual interest. In this video, we want you to dive into our world, giving yourself a clear insight into IFERP. But first, who are we and what do we do? Institute for Engineering Research and Publication, IFERP for short, is one of the world's largest non-profitable professional associations operating under the Techno-Elite Research and Development Association, TRADA, meant for research and development in the field of engineering, science and technology. We are one of the largest professional bodies for engineering professionals in India and across the globe, established in 2014. We extend to every part of the globe, with more than 28,000 professional members and 34,000 student members. Our growing membership at IFERP has led to an increase in members from Europe, Asia, Australia, Africa and the USA, fostering networking opportunities that strengthen ties within and across countries and technical communities. The mission of IFERP is to connect professionals at an integrated platform for growth to divert knowledge and skills towards the sustainable application of professional education and more. And our vision of digitalizing innovation processes through our professional networking services and more has pushed us forward to our achievements today. With our mission to make our academic services reach the top and grassroots level of institutions, we are funding colleges to organize conferences and scientific events, enabling researchers incubated at different areas of our country to directly contribute towards the transformation of India. IFERP simply connects engineers, exchange global innovation, and acts as a bridge between researchers and academics by organizing international conferences, world conferences, faculty development programs, webinars, seminars, providing membership, and more. Some of our conferences include WCASET Malaysia, WCASET Jakarta, ICAS ETAM APU Malaysia, and many more. We've also organised scientific events in association with renowned universities done in the past, with over 28,593 journal publication services, creating opportunities to get your research paper published in high impact factor journals such as Scopus Indexed, Web of Science, Google Scholar Index journals, ESCI, UGC approved, L Compandax, ISI, Chimago Index journals, and many more. To make our operations smoother and more perfect, we have been supported by our associated organisation, Association of Cloud Technologists, Global Association of Nanotechnology, International Association for Big Data Analysts, International Association for IoT, International Wireless Network Association, and World Association for Structural Engineers. We have also worked alongside several clients and partners both nationally and internationally like University Malaysia Sabah, APU Malaysia, UNJ Jakarta, MC University Thailand, Ajman University Dubai, PUP Philippines, IIUM Malaysia, ISU Philippines, FCPC Philippines, WCC Philippines SRM University, Galgotia University, Amity University, and much more government and private organisations in different countries. At IFERP, we believe that there's always a better way to treat professionals, and that is what we do. If you would like to see more or go deeper on IFERP, you can visit our website at www.iferp.in.
Hello everyone, good evening and welcome to our webinar on Biology in Concrete organized by Institute of Engineering Research and Publication IFERP and our academic partner United Innovators. Welcome all, and today for discussing on Biology in Concrete we are being accompanied by Dr.C.V.Siva Prasad sir. Sir, are you online? Hello, can you hear me?
Yes ma'am.
Hello, how are you? Good evening.
Yeah, good evening.
Welcome, sir, to our webinar. Sir, I guess your cam is off.
Okay, I will on. Yes.
Yes.
Good evening.
Good evening.
Thank
How are
you for.
you?
Yeah, I'm fine.
So, before we begin, I would like to introduce our speaker of the day, Dr.C.V.S. Prasad sir, and he's associate professor at Department of Civil Engineering at Vignana Bharathi Institute of Technology at Hyderabad from India. He has done his doctorate from ANU on bacterial concrete as a self-healing material and experimental investigation. Prior to joining in VBIT, Hyderabad, he was a lecturer in Bule Hora University in Ethiopia during the year 2015-17, and he has worked as structural engineer in Bosho Architects Private Limited at Hyderabad. Presently, he's a structural consultant also in Hyderabad, along with his teaching and research. And he's working for Unnat Bharat Abhiyan sponsored research projects with state and central government. His research interests are bacterial concrete, industrial RCC, and steel chimneys, special concrete structure materials. He has published one book on industrial RCC and steel chimneys. He has organized several seminar, workshops, FDPs during his tenure. He has published several papers in reputed journals and presented papers in international conferences and national conferences. He is also a reviewer for various reputed international journals. He is associated with various collaborations from different countries. Dr.C.V.S. Shivaram Prasad sir, he is also a life member of Indian Concrete Institute and Indian Institute of Bridge Engineers. Thank you, sir, for joining us today. Welcome.
Yeah, thank you so much.
Yes,
Thank you for
without any
accepting my
delay,
request.
we will move on to the presentation. It's an honor to have you, sir. Thank you
Okay.
so much. Okay,
Yeah.
and we'll move on to the
So thank you so
presentation.
much. So today topic is
Yeah, you may
okay.
begin, sir.
So yeah. Thank you so much. Now the today topic is biology in concrete. It is most important one for civil engineers, especially nowadays. Most of the cases, if you observe, the cracks are creating major problems in our buildings. Not only buildings, if you go for the bridges or any RCC structures, cracks are creating a major problem, especially for the leakage of water. So after that, the structure will go deterioration and also the strength is decreasing. So that's why there is an innovative technology that is nothing but biology in concrete, and some people they call it as a self-healing concrete, and also some people they call it as a bacterial concrete. Okay, it is very need of the hour, especially for the civil engineers. Okay, so before that, myself already introduced by Jejila. So here so my details are there. Yeah. So if you observe this picture here, whatever I show on the pictorial view here, maybe in your homes also, most of the people facing the cracks. So the cracks are creating most major problem, especially for the seepage of water, and most of the cases in rainy season also. The rainy season, it creates a lot of problems in our homes. Not only RCC structures, if you go with the bridges or canal structures, wherever the concrete structure is there, definitely after constructing within 2 or 3 years, the cracks are forming at different stages. So that's why to repair this one, we have a lot of techniques are available nowadays in our society, but the cost of those techniques are very high. Okay, next move on to another slide. Not only in buildings, if you observe in the rigid pavements are nothing but the concrete roads. So the concrete roads also, after some years of the construction, in most of the cases, if you see in village roads, those roads are made with the concrete roads. Those are nothing but rigid pavements. Those rigid pavements also, due to vehicle moving, are also load is more and more. So that's why there is a lot of creation of cracks are there. So once the cracks are occurred in the structure, definitely the strength of structure will decrease gradually. So it is very problematic, and also the construction cost is more. At the same time, we have to decrease the maintenance cost. So nowadays, once if you have to repair these type of the cracks, it is create a lot of expensive, and also not only expensive, again you have maybe there is no guarantee whether it will be permanent repair or not. After some years, maybe 1 or 2 years, definitely again cracks will occur. That's why we have to create one new technique, especially for sealing of these cracks or healing of these cracks. Maybe we know already different types of the cracks, like micro cracks or nano cracks, either autogenous cracks or inside crack, internal cracks or external cracks, whatever it may be. So the main motto of this research or this topic is a trending topic in civil engineering. Not only civil engineering, most of as a common people they must know regarding this research. Okay. So move on to this slide, that is a crack repairing system. This crack repairing system nowadays, whatever we are adopting these repairs, the first one is a stitching method, and second one is the epoxy injection, and third one is a cement grouting, and fourth one is a drilling and plugging. So these techniques are nowadays using for a lot of RCC structure, like RCC structures means buildings or bridges or any type of reinforced cement concrete structure. And the thing is that if you observe this one, definitely they need a labor. It requires a lot of material, and again the labor cost. So everything that it all are expensive. So that's why we have to come up with a new technique that is nothing but self-healing concrete or bacterial concrete. If you observe this one in the stitching method or epoxy injection or cement grouting or drilling and plugging, whatever it may be, those things are maybe expensive. We have to analyze the structure, how the crack is affecting on the structure, everything first we have to analyze, then only we have to adopt one technique, then we have to implement on that one. Nowadays most of the cases epoxy resins also we are using, and also we are using different types of the techniques, especially to repair this type of the crack. So everything is expensive. That's why we came up with a new technology that is nothing but bacterial concrete. Okay. So if you observe this diagram here, you see for the cracks, especially for the buildings or reinforced structure, whatever it may be, like bridges, there is a little bit problem even we can do. But if you observe a reinforced concrete structure in the swimming pool, when swimming pool is there, when the people are doing or enjoying with swimming, definitely if there is any crack, if you observe here, there is a stitching here. If you observe like this, the water will move from that one. Okay, the passage of water is more. So definitely it is creating lot of problem also. That's why the bacterial concrete is a need of the hour for the society. And also here, if you observe this pictorial view, generally we have different types. Whatever I explained still now, that one, the society, whatever the adopting techniques which we are using in our society. The first thing, why don't the concrete itself healing? That means self-healing of that concrete. We have to think about that. That one, if you observe this one, there is a concrete cube is there or concrete material is there. Why don't it heal itself? For suppose if you observe your body, whenever the wound is occur in your body, after some we will apply some any treatment. Okay, but after some wounds are automatically it will heal. Same like that, why don't we apply the same thing in our concrete? It is maybe innovative, but definitely it is doing most of the countries they are following. Even in India also, in Hyderabad we are following this one. Okay. Next, what is the need of bacterial concrete? Why we are using? If you observe the recent days, the bacterial concrete has been emerged as a remedial measure of healing cracks in structures like bridges, RCC buildings, RCC pipes, and canal lining and pavements. Again, in the pavements, we have rigid pavement. Definitely, it is emerged as a remedial measure. That means it is a remediate the cracks. Okay. Not only that, the crack formation is a incredibly common occurrence. Everyone knows that, in a concrete, in your building, definitely in your home also. If you observe that one, so whenever the crack formation is arrived in your structure, it permeates the water, and completely there is a different sort of chemical into that concrete through that cracks and reduce the strength. So whenever the strength is decreased, automatically other things, the durability also will decrease. So additionally have an effect of reinforcement also. There is a reinforcement, whatever we are using in beams or columns or slab, whatever it may be, definitely there is a reinforcement is there. So again, it will affect on the that reinforcement. Once it will affect on the reinforcement, there is a possibility of corrosion. Okay. For repairing the cracks occurred within that concrete, it needs a regular maintenance and special sort of treatment, which can be terribly expensive. I told you previously, the all things whatever we are doing present days, those all are expensive. So there are so many bonding agents are used in the construction industry to heal this type of the cracks. Those are latex emulsion and epoxy bonding agents and surface treatments with silence and also siloxanes, but these are harmful to the environment. Okay, hence the bacterial concrete or a self-healing concrete, it can be more economical and also durable solution to mitigate the cracking of concrete structure. That's why it is a need of the hour for the society. Okay. And next, if you observe this one, how we will do how the biology is occurred in the concrete. For suppose if you observe here in this diagram, the concrete mixture, whatever it may be, in the concrete mixture we have how to incorporate the bacteria. And again, the bacteria also we have different ways, different types are there. Those are maybe Bacillus subtilis, Bacillus pasteurii, and also lot of bacteria are there. In another slides I will show you definitely. So whenever the bacteria is incorporated in that one, again there is a mechanism is there, and also there is a microbiology activities are there, and there is a chemistry and hydration process, and each and everything is there. So I will explain clearly. So this is the type of bacteria, and also this concept where it was started first. So coming to this self-healing concrete, this concept, especially the bacterial concrete concept, was started or introduced by Henk Jonkers. Before Henk Jonkers, there is a professor from South Dakota University from USA. There is a Ramakrishnan professor. He also started, but this is not implemented more. But Henk Jonkers, who is the professor from Netherlands, the university is Delft University of Technology. This is a novel technique is adopted in remediating cracks and fissures in concrete by utilizing the microbiologically induced calcite precipitation. We call it as a MICP. That is nothing but the technique. We also call it as a biomineralization. These all techniques we call it as a biomineralization or bacterial concrete or self-healing concrete, whatever it may be. The major important thing we must know about the how the calcium carbonate, that is nothing but limestone, how it will form in between the cracks. That is a major technique. Okay. In this study, whatever we are going to explain in this study, the experimental investigation have carried out to arresting the cracks in the concrete using Bacillus subtilis bacteria and calcium lactate. The complete research I used the bacteria that is nothing but Bacillus subtilis. It is a family of Bacillus. Along with Bacillus subtilis for the food of that bacteria, I used as a calcium lactate. Okay, both are most important ingredients of, along with ingredients of concrete design, whatever it may be. The ingredients, like we know already, the concrete ingredients: cement, fine aggregate, and also coarse aggregate and water. But here I am using, along with those ingredients, I use bacterial concrete. In the bacterial concrete, I used Bacillus subtilis bacteria and also calcium lactate as a food for that bacteria. Okay, here the Bacillus subtilis bacteria with calcium lactate is used in different percentages, such as 5%, 10%, and also 15% for of cement weight for two grades of concrete. The first one is M20 grade concrete and also for M40 grade concrete. If you observe, generally we use the grade of concrete in normal structures in our town or in our city. For up to G+5, generally we use M20 grade concrete. But if you observe for the rigid pavements, that is nothing but concrete roads. For the concrete roads, generally we use from M30 grade. That's why, to know that for the higher end, I use the M40 grade concrete. That's why both grades are most appropriate, and also practically, if you observe this one, it is very important. For this research, I used both M20 grade concrete and also M40 grade. Okay, with the river sand mixes and also crushed stone sand mixes as a replacement of fine aggregate are considered in this research. Why I used crushed stone sand? If you observe most of the states, the scarcity of river sand, because there is no sea, coastal area. So those cases there is no rivers. So at that time we didn't get the river sand. That's why we have to go with the manufacturing sand, or we call it as a crushed stone sand. That's why I used for the entire research, especially along with the river sand, I used crushed stone sand. It is the cost also very less. If you observe this one, I used river sand along with the crushed stone sand of replacement of fine aggregate in the M20 grade concrete and also M40 grade concrete. So we used both grades of concrete. And here, how the experimental investigations are carried out to study the effect of bacteria. Everyone knows whatever the concrete we are using, first we have to do all the research in our laboratory. So that is nothing but I used the research on bacterial concrete, especially on fresh concrete and also hardened concrete. We know that the fresh concrete, the slump cone test or compaction factor test and VB test, whatever it may be. So everything we done, especially on hardened concrete, I performed the test on compressive strength and split tensile strength and flexural strength of concrete. These three are most important to know the mechanical properties of concrete. So here the physical properties everyone knows, and also along with physical properties of cement, whatever we find out, definitely we have to calculate the mechanical properties of that concrete. So for this case, as per the code book, as per the IS 456 and also as per the IS 586 or 5816, there are different types of the Indian standard codes are there. To know the compressive strength and the split tensile strength and flexural strength of conventional concrete along with the bacterial concrete, I used there is a code book that is IS Indian Standard 516 and also 5816. Along with these two code books, I used 5816 also. Especially for the mixed design of code book, I used IS 10262 code book. This is a code book with 2 years, that is IS 10262 2009. Along with recently there is a code released from the Bureau of Indian Standard, that is 10262 2019. So here, especially when you want to know the compressive strength of concrete, the curing period may be at 28 days. Okay, but to know the exact value, why? Because the bacterial concrete, it is a sealing of concrete. That means healing of concrete. So to know that how much it is seal the concrete, how the bacteria, the microorganism is living in that concrete, everything whatever we are doing the research, I done the curing period are the ages of 3 days, 7 days, 28 days, 90 days. Especially for the compression strength purpose, I used up to 365 days also. But later, whatever I done for the non-destructive testing, everyone knows as a civil engineers, non-destructive testing is nothing but rebound hammer method and also ultrasonic pulse velocity test. So this ultrasonic pulse velocity test I used to measure the quality of concrete. Okay, so at the ages of 3 days, 7 days, 28 days, 60 days and 90 days in different mixture using the cube specimen of plain and also crushed stones and bacterial concrete. Okay, after that, those are the mechanical properties and also non-destructive tests. So but if you want to know why, whenever we use this bacterial concrete for the roads, definitely you must know the abrasion resistance. That is the abrasion resistance. For suppose there is a road, when you move a vehicle on this one, definitely it needs there is some friction. So for that purpose, we must calculate the abrasion resistance of that concrete. But generally, whatever we are using M30 greater than M30 grade or M40 grade of concrete, generally the abrasion resistance is good. But we are implementing or we are incorporating the new technology, that is a bacterial concrete. We don't know how much the abrasion resistance is there. That's why there is a test as per the ASTM standard, that is nothing but Cantabro loss test. That Cantabro loss test I used for the bacterial concrete. It is a very good test, especially the new test. Maybe if the researchers are there, you can use this Cantabro loss test for your research. Okay. So after that, the main agenda of this research is you have to know the microstructure of bacterial concrete. That is the microstructure of bacterial concrete. Whenever you have to know the microstructure of the concrete The first and foremost thing is the scanning electronic microscopy and also energy dispersive X-ray analysis or X-ray dry fraction test. We have different types of the tests are available, how the elements are available in the concrete. So to know that one, and also how the calcium carbonate, that is nothing but limestone, whether it is formed or not, if you want to know that, definitely we will do the microstructure analysis, that is nothing but scanning electronic microscopy and also energy dispersive X-ray analysis, XRD test. So these both are very critical tests and also very difficult to perform this test. There is a lot of procedures is there, and the equipment is available in very specific locations. Okay, so move on to another slide. First of all, what is the bacteria? So as a civil engineers, we don't know what is the microorganism, microbiology, we don't know chemistry, maybe a little bit we know at the time of hydration of cement or hydration of concrete. But along with this bacteria, it is very difficult. You must know the microbiology, and you must know what is the different types of the bacteria available, whether they are used for the concrete or not. So everything is different. So now, what is the bacteria? So if you observe, bacteria are microscopic, a single-celled organism that exists in their millions in every environment, both inside and also outside other organisms. Some bacteria are harmful, but most serve as a useful purpose. If you observe this one, most of the cases there are useful bacteria are available. Okay, they support many forms of life, both plant and also animal, and they are used in industrial and medical purpose processes also. Next, what are the classification of bacteria? So how the what are the different types of the classification is there? Based on shapes, based on ground strain, and also based on oxygen requirement. We have different categories are there. Based on shapes, like rod-shaped bacteria is there, and spear-shaped bacteria is there, and spiral-shaped bacteria is there. Based on ground strain, there is a gram positive and also gram negative. And based on oxygen requirement, we have two types, that is aerobic and also anaerobic. And most of the cases, as per the literature review and as per the scientists, as per the experts of in this field, and even if you observe the previous literature review also, if you observe the Bacillus subtilis or Bacilli family, it is a more strength to observe this one, and also to create a calcium carbonate. Okay, this calcium carbonate, this is nothing but limestone. So there is a limestone possibility of creation in between the cracks. Definitely it is possible of Bacillus subtilis bacteria only. That's why everyone is choosing from Bacillus family. But again, in the Bacillus subtilis, different types of bacteria are there, like Bacillus subtilis and also Pasteur. If you observe here, we have different types of the bacteria used in the concrete as a from Bacillus family. That is nothing but Bacillus pasteuri, Bacillus species, coli, and subtilis. It's the most important thing. Even I use the research in this case, Bacillus subtilis, and also Bacillus cohini, halodrums, and etc. So most of the cases to seal the cracks, especially we are using Bacillus subtilis. It gives immense power and also calcium carbonate, that is nothing but limestone. Okay, moving to next slide. So this is the Bacillus subtilis. There is a different procedure is there, later I will explain. So first, before going to that, as earlier I told you, Bacillus subtilis, which one, it is the most available bacteria, it is not harmful, and it is create calcium carbonate within the concrete when you add along with the cement, fine aggregate, coarse aggregate, and water. So there are some ingredients are there along. When you add this Bacillus subtilis bacteria, definitely there is a possibility of sealing the cracks. So how we will do that possibility of sealing the cracks? When we use the ultrasonic pulse velocity test, otherwise we use the microstructure analysis. I will show you the diagrams also. Yeah, these are the types, and everyone knows as a civil engineers, there is a possibility of water cement ratio. If you observe or if you create a water cement ratio as per 10262 2019 code book, if you observe that one, there is a, if you add more water, definitely there is a loss of compressive strength. That's why water and cement as per the code, if you follow that one, definitely there is a strength is more. Otherwise it is difficult to maintain the compressive strength after some years. That's why most of the structure nowadays creating the cracks, or also nowadays their deterioration also is more, and durability is not good, and also lifespan is very less. Okay, that's why while preparing the mix of concrete, whatever we are using the grade, we have different types of the grade, that is M20 grade, M30 grade, M40 grade. Even for the high rise structure, most of the cases we are going for the high grade concrete also, but basically we start from the M20 grade concrete. If it is a rigid pavements, definitely we start with the M30 grade or M40 grade concrete. That's why whenever if you prepare a mixed proportion, you must follow code books as per the Indian standard and Bureau of Indian Standard code books. You must follow that one. If you are a foreigner, definitely you follow from your code books. If you are from USA, you have to follow ASTM codes. If you are from Europe, you have to follow Euro codes. Like that, every country having their own code books, especially for the creation or also for the proportion of concrete, and how the chemical reaction will occur within the concrete, which is the most important thing, as earlier I told you. So especially while we add the concrete, whatever we are in the ingredients, we are adding the food for the bacteria, that is nothing but calcium lactate. There is a process I will tell you. Once if you add these ingredients, whenever the water is sprinkling on this concrete, or whenever the oxygen is entered into that concrete, when there is a small holes are available, that is nothing but we call it as a voids in the concrete, or whenever there is autogenous cracks or micro cracks, whenever the cracks are like that, when moisture is entered into the concrete or water is entered into the concrete, once it is entered, definitely that bacteria will germinate and it will eat the, or it will consume the calcium lactate around that bacteria. Then it will release the calcium carbonate. That is the main mechanism in this bacterial concrete. But if you want to know the chemical reaction of this concrete, that chemical reaction, the calcium lactate, which will react with the oxygen, whatever which it is available in the concrete, it will form the calcium carbonate and the calcium water. So again, the calcium carbonate react with the calcium hydroxide. Everyone knows the calcium hydroxide will form at the time of hydration of cement. So at that time, again, it will react with the CO2, it will form the CaCO3. That is nothing but calcium carbonate. Okay. Yeah. What is the advantage of this bacterial concrete? If you observe, there is a lot of advantages are there. Okay. The first one is it will improve the compressive strength. Not only compressive strength of concrete. If you, as per my research, along with compressive strength, there is a possibility of increasing split tensile strength and also flexural strength of concrete. So again, there is a better resistance towards freeze throw attack reduction. Whenever the strength is more, there is a possibility of decrease in permeability of concrete. That means there is a, whenever you pour the water, the permeability will reduce. That's why if you use this bacterial concrete, there is a possibility of decrease in the permeability, and there is a reduction in corrosion of reinforced concrete, and also it is eco-friendly. These are the advantages of bacterial concrete. Next, this one, the objective of this research, entire research, whatever I am going to explain, I told you previously, I studied it is effect of fresh concrete and also hardened concrete, and we have to investigate as a crusher action as an alternative to the river sand. I think everyone knows recently in Andhra Pradesh state, if you observe, in the state there is scarcity of river sand. At the same time, the government taken a new policy. So at the time of government formation in Andhra Pradesh, there is a lot of scarcity is there. They stopped the river sand. So at the time, lot of builders they they didn't get the river sand. That's why they used most of the cases crushed rock sand. It is nothing but M sand. So the properties are very same. Okay. Next, to study the physical durability of bacterial concrete, that is nothing but physical abrasion resistant test I done as a Cantabro State. It is very useful for your research. Okay. And next, know the bacterial concrete microstructure. I told you already, that is SEM analysis, X-ray analysis, and also EDAX. Okay. Next, to evaluate the dynamic modulus of elasticity. Everyone knows we are doing modulus of velocity of a cylindrical portion. When you do the cylindrical portion, the time of split resultants, we calculate the modulus of velocity of concrete, especially for the conventional concrete. But we are doing here for the bacterial concrete also, but not that test. I followed the ultrasonic pulse velocity test. This is very important test. Okay. Next, coming to the characterization of a material. Whatever we are doing research, the characterization of the materials are most important. If you don't know the physical properties of your material, if it is very difficult how the chemical reaction will occur or how the hydration process will happen in our concrete. Okay. So for that purpose, if you observe here, the cement whatever I use, that is nothing but ordinary Portland cement. The grade of the cement is 53 grade concrete. So at the end research for the concrete, especially I used 53 grade concrete. It is nothing but OPC. Most of the cases in the society we call it as a OPC. For generally plastering purposes, we used 43 grade and also 33 grade. But nowadays no one is using 33 grade. But plastering purposes, some people are using the 43 grade also. Okay, for the entire research I done already for the 53 grade of cement. Okay, and the fine aggregate, whatever I use the fine aggregate, I told you earlier, as a replacement of the river sand, I used crushed rock sand, and I done the specific gravity of cement is 2.68 and also 2.77. And coming to the coarse aggregate, the coarse aggregate we used the crushed granite broken stone of 20 mm nominal size. Generally, we use the 20 mm along with the 12 mm chips. Okay, in the society, in our construction activities also, most of the cases we are using 20 mm. It is a mixture of 12 mm also. And coming to the specific gravity of that coarse aggregate is 2.71. And coming to the water, so here the water is locally available potable drinking water used for the entire experimental work. So I told you characterization of materials are most important. If you do with the 43 grade concrete, you didn't get the strength properly. That's why I used the 53 grade concrete. Okay, as per the code book IS Indian Standard 4031 1996. And coming to the bacteria, how we will develop a bacteria and how the microorganism is developed. So that culture of microorganism, everything I done in the Hyderabad, that is there is a BioLife company, that is a DBS BioLife Private Limited Laboratory. So how we will maintain this culture of the bacteria. So the culture was isolated in the DBS BioLife Laboratory, and it was maintained on nutrient agar slabs constantly. Constantly we have to maintain that one. The irregular white colonies. After some days, there is a formation of white colonies on the nutrients agar. So there is a formation as like a white colonies. So when we require a colony of that culture, so while performing or while incorporated to the concrete, we have to take in a single colony of culture. It was inoculated into the nutrient growth of 25 ml in 100 ml conical flask. At the same time, this setup was maintained at 37 degrees centigrade temperature, and also we placed on orbital shaker at 125 rpm. This is the most important thing while performing the culture of bacteria. If you don't done all these things, it is very difficult whether the bacterial survival or not. That's why the culture of bacteria is most important. Along with this culture of bacteria, the medium composition required for both of culture, we used peptone, NaCl, and yeast extract. So the peptone is 5 grams per liter, NaCl is 5 grams per liter, and yeast extract is 3 grams per liter. So this is the culture of bacteria. So I told you we done in DVS BioLife Private Limited, Hyderabad. So whenever we need a colony, definitely we take a single colony of that culture. We have inoculated into the nutrient growth of 25 ml in 100 ml conical flask. At the same time, this setup is maintained at 37 degree centigrade temperature. We placed in orbital shaker at 125 rpm. Okay. So now coming to the calcium lactate. So I used calcium lactate to the entire experimental work. So along with this Bacillus subtilis bacteria as nutrient growth. So it is the form of powder having white color. Even you can find this calcium lactate in most of the outside shops. Otherwise, you can order it is on online also. It is available available as a 250 grams. Next, if you observe these diagrams, this is the most of the cases whatever I performed in the laboratory. This is the bacterial solution. Okay, and here if you observe, this is the bacterial solution. After whatever I told in the previous procedure, finally we done like this. This is the solution we used as a 5% and 10% and 15% for the cement weight of concrete. Whatever we prepare for the things, either it is M20 grade concrete or M40 grade concrete. And if you want to do, finally we kept as in like this. We have different types: vectobar, baseless, pasturi, and this is a baseless subtilis. Okay, and these are the types of the mixes. So how we perform this type of the mixes? I told you we are using different type solution. The bacterial solution, that is a 0% of bacterial solution, that is nothing but conventional concrete. At the same time, 5%, 10%, 15% of bacterial solution. So I performed for the RBC, that is nothing but bacterial concrete with reversal. At the same time, CBC, that is nothing but bacterial concrete with conventional concrete. So here, if you see here, the mixed design I done as per 10262/2009 code book, as for the M40 grade concrete. So here, one is cement proportion, 1.65 is a fine aggregate, 3.23 is the coarse aggregate. This is the most important thing, especially to perform or to create a concrete of grade. And this is the workability test. As I earlier told, if it is a workability of concrete is good, then only we will perform this concrete in the site. Especially there is a possibility of workability. If you didn't get the workability, good workability of concrete, there is no passage of this concrete in the reinforcement. Whatever we do for the slabs or columns or beams, whatever it may be. So the workability test is most important. For that purpose, the workability test I use both for river sand mixes, crushed stone sand mixes. The workability I got 58 mm and also 55 mm. I performed in our laboratory. So once if your workability is not good, when it is a zero slump or two, it is not good for the passes and also passes. That is nothing but flowability of concrete. Whenever the flowability of concrete is not good, we don't use that type of concrete for the construction purposes. That's why I performed this one on slump as a slump cone. This test we call it as a slump cone test. So it is very good and we got good results. This is all preparation of bacterial solution. Okay, so this one I told you earlier, there is a calcium lactate. So that calcium lactate, whatever it is in white color form, even I added here like a 5 grams, 10 grams, 15 grams, same like whatever the percentages. So once it will change the color to brown color, so we have to add at the preparation of your concrete. Yeah, even I done before going to the microstructural analysis and mechanical properties of concrete, I performed a good thing here. Maybe it is innovative. Why? Because I taken a cube at the time of casting of concrete cube. We have a mold, concrete mold. The size is 150 by 150 mm. So at the time of casting, I created a crack at the fresh concrete at the time of placing in the cube. Okay. So after that, what happens? After 24 hours, we dismantle the cube and we put for the curing purpose. So I observe this crack after 28 days. How it will develop? I will show you next diagram. You see, there is some white formation that is nothing but calcium lactate. If you observe the previous slide, the crack is more. If you observe this slide here, the grade of concrete is M20 grade. The almost crack at the time of 28 days, it was healed. Okay, it was sealed. Why? Because we incorporated the bacteria, that is nothing but Bacillus subtilis. At the same time, along with bacteria, I used calcium lactate. So there is a possibility of this one. That's why I performed all the tests up to 365 days. And also I done microstructure analysis, that is a scanning electronic microscopy and all, especially to know whether the elements are formed or not. Okay, that is nothing but CaCO3. Yeah, this one also. Here, if you observe here, I created it here 2 cracks. Yeah, this is the most important one from the Young Jonkers. If you observe here, the crack, but after sealing of cracks automatically, so it will look like this. This is the most important phenomena. Whatever we are doing here, so in this research, especially whatever we are doing here in the laboratory, the crack formation, how it will seal, and how what is the mechanism, and how the calcium lactate, that is and also preparation of nutrient growth at the time of microorganism. So everything is important. So successfully we are doing this one, and nowadays when we are applying for research fund also for this purpose. Most of the cases in Hyderabad, some companies they are giving funding for this concept, especially it is a need of the hour for society, not only for the civil engineering research. Every human being need of this concept. Why? Because everyone is living in homes. So if the cracks are occurred or vulnerable, it is definitely there is a possibility of destruction of your structure, okay, or collapsing of your structure. That's why it is the most important one, that is a self-healing concrete. Yeah, if you observe here, there is a control specimen and bacterial concrete, how it will heal. Yeah. This is the curing tank. Once if you dismantle your all your cubes or whatever the cylinders or beams or prisms, we have to curing up to 28 days, okay, or 90 days, 60 days, 160 days, 180 days, or 265 days. But I done everything up to 365 days. That means 1 year. So 1 year for curing in your same tank is very difficult. I removed at every 10 days, 15 days. I entered already. We poured the fresh water for curing purpose. This is a very important thing. And also these are the specimens ready for the structure for the compressive strength, and also speed tensile strength for these specimens we used for the to know the strength characteristics. That is nothing but mechanical properties. So here the methods how we done, that is the compressive strength, split test, flexural test, Cantabro loss test, and UPV test, and same EDAX XRD analysis. And coming to this one, the strength properties, the variation of components of bacterial concrete with curing age of river sand, at the same time crushed red sand. If you observe these diagrams, the first one is the M20 grade concrete for the river sand. The second one is M20 grade concrete for the crushed sand mixes. Same like that here, M40 grade concrete and also M40 grade concrete for river sand and also crushed rock sand. If you observe here, there is a possibility of increasing at every level. If you observe here, the first diagram, the first graph, that is M20 grade concrete, it is nothing but at 7 days, 14 days, 28 days, 60 days, 90 days, 180, 270, 365 days. I taken the specimens, I casted the cube, same like that. We done experimental work up to different stages from 7 days to 365 days. There is a possibility of increasing at every level. That means why it is possibility of increasing strength means whenever the bacteria or calcium lactate adding into the concrete, there is a possibility of increasing the reducing the permeability of that cube or structure, whatever it may be. So whenever the permeability decreases, that means there is no voids. Definitely the voids are not there. There is a possibility of increasing compressive strength. Even for our research also, if you observe here for the 2 grades, there is a increasing in the strength. Okay, same like that, the split tensile strength also increased from 7 days to 365 days, especially the M20 grade and also M40 grade as a replacement of fine aggregate, while we are using the river sand, but here we use crushed sandstones also. That is nothing but M sand. Same like that, the flexural strength. The flexural strength we perform in UTM. Okay, so in the UTM, Universal Testing Mission, so we done as a beam, we casted a beam that is the size is 10 centimeter by 10 centimeter by 50 centimeter. That is nothing but 100 mm by 100 mm by 500 mm. If you observe our cube strength, we casted a cube 150 by 150 by 150 size, and the cylinder is 150 diameter and also 300 mm height of the cylinder. The same thing we casted and also we dismantled that, and we kept it for curing purpose up to different stages from 7 days to 365 days. Then we performed a test on compression split and flexural test on hardened concrete. We got some results. Definitely there is a possibility of increase in the strength. Next, coming to the abrasion resistance of bacterial concrete by using Cantabro loss. If you observe this test, generally this Cantabro loss test, we call it as a the equipment, the Los Angeles test, generally as per the IS code books, that is the Indian Standard Code books, we use this test for the aggregates. But as per the ASTM code book, American Standard code book, whatever it may be, ASTM, as per the ASTM code book, we use this Los Angeles test, the Los Angeles equipment we used for the Cantabro loss to know the physical abrasion of the concrete. So for this purpose, we maintained around 32 revolutions per minute, and we casted different types of the cubes. That is nothing but 100 mm diameter, 150 mm diameter, and also 100 mm height of a cube, the height of a specimen. That specimen, we casted the 3 specimens. So generally for the aggregates, whatever we are using this equipment, we use some balls, iron balls. But in the Cantabro loss, we removed that balls. Now we placed 3 specimen into this equipment. We will close that one, the door, whatever it may be, the upside in the shown in the equipment. So we will rotate this one from the mission. So once if you switch on that one, the rotating rate, we take in 32 revolutions per minute. Okay. Yeah. Here these are the specimens. If you observe this one, these are the specimens here. The diameter is 150 mm and also height is 100 mm. So it is very important. No one done the research on this one, the preparation of the molds like this, especially Cantabro loss test. Some people they done on the permeability concrete, that is the Kyodong, and Professor S.K. Rao, especially they done on roller compacted concrete. But the bacterial concrete, I am the person to perform this test. Yeah, these are the specimens we casted. 3 specimens, especially for the river sand mixes and also crushed stone sand mixes. Those are BC5, BC10, and BC15. That is nothing but 5%, 10%, 15% of bacterial solution of cement weight. So we casted this one at the every revaluation. At every 50 revaluations, we will take out and we will clean that one. We will note down the value. Again, we will put in our, we will kept in that mission, and we will switch on the equipment and we will perform 32 revolutions per minute. So after 50 revolutions, again we will take out and we will clean whatever the debris are available inside. We will remove. Again we will put. Same like that, the process we will do done up to 300 revolutions. Once if you do this one, if you observe this pictorial view in the images, the first images is the specimen of 24 hours. That means after the casting. So after 28 days of curing. So but after the kept in the mission, it will rub each other. So if your concrete is not good, that means your quality is not good. Definitely that they collide each other, they collapse. That means your concrete is not good. The abrasion resistance we will perform, especially for the roads, that is nothing but concrete roads, rigid pavements. Once if you don't perform this rigid pavements test on this one, that is a Cantabro loss test, it's very difficult. Okay. Yeah, these are the specimens before placing into the mission. The Los Angeles test, the Los Angeles equipment, and here after 50 revolutions, after 100 revolutions, how the specimen look like. After 150, 200, 250, 300. That is enough as per the ASTM. Okay. So these are the tests. Whatever we done, the number of revaluations from 0 to 350. That is from 50 to most of the cases 300. If you observe this one, the revaluations are most important. We performed for 3 days, 7 days, 28 days, and also 90 days. Only up to 90 days only I done. So even here also revaluations increasing. Number of revaluations, the Cantabro loss is also increasing. Same like that, I performed both grades, M20 grade concrete or the same time M40 grade concrete. Okay. Yeah, here. Number of revaluations of river sand mixes, same like that. sorry, yeah, river sand mixes and also crushed stone sand mixes. So the Canterbury loss at 28 days for the conventional mix with river sand is 11.36%, with increase in bacterial solution from 0% to 15%. The Canterbury loss is decreased up to 10% solution. This is due to fact that there is a presence of calcium lactate is prominent. Next, the decrease in Canterbury loss at 3 days, 7 days, 28 days, 90 days are 23.6% and 28.28%, and also 34.4% and 47.96% respectively for the river sand mixes. Same like that for the crushed stone sand mixes, there is a decrease in Canterbury loss or 37.29%, 23.26%, 23.75%, and 48.24% at different ages like 3 days, 7 days, 28 days, and 90 days. And next, most important test, that is the ultrasonic pulse velocity test on bacterial concrete. It is also non-destructive testing, same like a rebound hammer method. Everyone knows rebound hammer method. Okay, here by using the ultrasonic pulse velocity test, we know the quality of concrete also, and we will calculate the compressive strength of concrete, and we will know the crack, how the crack was formed, what is the size of the cracks formed, everything. Okay, so here if you observe here, I performed the test. Generally the ultrasonic pulse velocity test we performed at the one edges, that means two edges we will put the transducers, and we will take the wave, how wave formed inside the concrete. Based on the code book whatever it is given, we will calculate the quality of, we will check the quality. But how I done? So I taken three facets of concrete. The first facet is F1, second facet is F2, third facet is F3. I performed three facets. So based on these three facets, we taken some values. So as per the, we check with the code book values, so whether the quality of concrete is good or not. I will show you in next slide. Yeah, this is the test I performed in the laboratory. Okay, this is the equipment of ultra UPV. Most of the cases we call it as a UPV test. Directly as a short term, it is a UPV test. We performed on the concrete cubes. The size is 150 mm by 150 mm by 150 mm. Okay, this is a test. Yeah, if you observe this one, the quality of concrete as a function of UPV, there is a code book that is nothing but as per Indian Standard 13311 Part 1 1992. So the pulse velocity crossing probing is in kilometer per second. If it is greater than 4.5, the concrete quality is good. If it is less than 3, definitely it is a doubtful. We don't use that concrete for the construction purpose. So if it is in between 3.5 to 4.5, it is good. If it is in between 3 to 3.5, it is the medium. So that's why most of the cases we used if it is 3.5 to 4.5. In some cases we opt for the 3 to 3.5 pulse velocity crossing also. So that's why if it is doubtful, we don't use that type of the concrete. And coming to the next slide. So here, if you observe the results, whatever we got from the experimental results, especially for the M20 grade concrete and also M40 grade, but we performed the concrete mix on RBC from 0 to 15%, that is the bacterial concrete with river sand. Same like that we done for the crushed rock sand, and we call it as a M sand or crushed stone sand. So here the three phases, that is a F1, F2, F3. So I taken the average, and same like that it performed up to 3 days, 7 days, 28 days, and also 90 days. If you observe here, some cases the mix it is failing. Some cases it is very good. Okay, and I will show you in the next slide how it is formed in the not only numerical. Here if you see the time at 3 days, RBC 0 is doubtful, RBC 5 is doubtful, RBC 10 is doubtful, but at 90 days or 60 days all are good. Some cases especially crushed sand mixes it is excellent also.
Excuse me, sir.
Yeah,
so sorry to interrupt, sir, your slide is not visible actually.
Now
no sir.
Yeah, please check once, ma'am. Now is it visible, ma'am?
I think there is a network issue problem.
Okay.
No, so the slides are not visible actually.
Yeah, from which slide, madam?
From the last 2 slides.
Okay, okay. So now is it good? Is it visible, ma'am?
No, I guess that's because of the network issue. You may continue, sir. Maybe in between we can get back to the slides. It will be
Yeah, here
visible.
from my network is good, madam. No issues.
Okay, I
Okay.
guess. Okay.
I will continue. I will finish. Okay. Thank you. So later, even if you observe for the M40 grade concrete also, so both the things whatever we explained. But now coming to the scanning electronic microscopy analysis, the scanning electronic microscopy also same to know the calcium carbonate how it was formed. Okay, so especially for the same analysis, I explained the magnification factor like this from 1,000 to 30,000, and voltage is 20 kV. And for the conventional concrete, and also if you observe the bacterial concrete, there is a formation of calcium carbonate. Especially, we will do the calcium carbonate. Once if the calcium carbonate is performed, there is a possibility of healing of cracks. Okay. Same like that, I performed the EDX also. These are all pictures whatever I showing here. So all things are especially for the formation of calcium carbonate, it's nothing but limestone. Okay. Even for the EDX also, we will do for the number of iterations. So once if you want to know exact location, we will perform at the location, and everything I performed at the BITS Pilani Hyderabad campus. Same for the conventional concrete and also bacterial concrete. So it was the formation of calcium silicate hydrate, that is CSHL, and portlandite CH, and pores were observed in the all the concrete samples. In case of bacterial concrete, the precipitation of calcite was visible in the pores. Okay, so from the conclusions, whatever I am going to tell you are the 10% both for the river sand and the crushed stone sand. The mix is good, and it is suitable for the construction, and even for the M20 M40 grade concrete for the physical durability also good at 10%. The microstructure analysis is demonstrated. There is a possibility availability of calcium carbonate after 28 days. Okay, and there is a case study also done in Netherlands. Especially there is a field performance of bacteria based repair system. This is a pilot study done by Hank Jonkers. Okay, so they taken the everything this bacterial concrete applied for the parking areas. So after that the concrete was healed, so it was a patent one. Even for the roofs also it was a healed concrete. Okay, and thank you so much. This is from my side. I think everyone, thank you for attending this webinar. Thank you for giving opportunity of IFERP. Thank you so much. If you have any doubts, you can ask me.
Thank you, sir. Thank you for that informative session. It was not only informative for the researchers, but also for a common man, the common things that we have to know. So it was very interesting. Yes, sir, we do have some questions. The first one, it goes like this. Can it stop the seepage of rainwater through the rooftops?
Okay. So from the rooftops, maybe we will stop once if we use this bacterial concrete. Yes, we will stop. Why? Because we are doing Bacillus subtilis along with the calcium lactate. So generally, what we use for the construction process, we use the there is a Dr. Fixit is there, and also epoxy resins are there. Generally, we are using other roof places, but if we use this technique, definitely it will stop the rainwater also.
Okay. Thank you, sir. And the next question is, can we apply this product on old house to avoid water hold in walls?
Yeah, the thing is that for the walls is very difficult. Why? Because the walls we started inside the walls is the brick is there. Okay, but we are applying this one for the concrete. So generally we apply for the slabs and also columns or beams.
Okay, so I hope the person who have asked the question has received the answer, and thank you, sir, for answering the questions, and thank you all who have joined us today, and thank you once again, Dr. Prasad, sir, for presenting this wonderful presentation. And thank you to our academic partners, United Innovators, for joining us on this webinar. If we have researchers or students who are listening to us in this session, do visit United Innovators. They have amazing opportunities and assistance for PhD students, such as thesis writing and research article writing, as well as publication in reputed journals. So do visit United Innovators. We have given the link in the chat box. And further, if you have any questions related to the certificate downloading, do write to us at webinar@iferp.in. But the procedure is same like all the time I have been saying. Please do log into your IFERP account and download it from the certificates and rewards section. Or you can see it right on top itself, download the webinar certificate. So for more of upcoming webinars, do follow us on IFERP Facebook page, Twitter, LinkedIn, and Instagram, and do subscribe to our YouTube channel as well. So thank you all who have joined us in the live webinar, as well as those who are watching us live on YouTube as well. Thank you so much. Have a nice day. Good evening to all. Thank you, sir.
Thank you. Thank you. Thank you so much for giving this opportunity.
You're most welcome, sir.
Thank you.